A very warm namaste to all of you, and this is a super cool and super awesome session on the frequently recommended topic, that is application of biotechnology. Lecture number 1, where we'll be discussing about the very first topic from the chapter, that is the basis of biotechnological application as well as its importance in the field of agriculture. Clear? Okay, so this is a very interesting chapter where the children usually asked to take the class upon. Okay, that's the main reason why firstly I made the slides and just started the class. Okay, so a very interesting chapter and it's a scope for future job. So please study the chapter with great interest and please try to concentrate so that it may help you really in your future. Okay, so without wasting our precious time, let's begin our chapter, applications of biotechnology. So, biotechnology deals with the industrial scale production. Clear? So, biotechnology deals with the industrial scale production of biopharmaceuticals as well as biologicals. How? By using genetically modified microorganisms. You can either use genetically modified microorganism, genetically modified fungi, genetically modified plants, or genetically modified animal. Okay, so these genetically modified organism, you may be calling it as to be what? Transgenics. Okay, transgenics you may call it as, where their gene chemistry is being manipulated or their DNA sequence is altered by manipulation. So the very first plant, transgenic plant, was tobacco. The transgenic plant was tobacco. And the transgenic animal is the mice. Okay? The most of the transgenic organisms or animals are the mice. So what are the uses of these transgenic organisms? We'll be discussing in the upcoming session. Okay, so let's continue. So it deals with the industrial scale and not with the home. Okay, it must be in a large quantity and not limited to the homely environment. So the different applications of biotechnology are therapeutics. Okay, diagnostics. In the diagnostic technique, what all things in the PCR, polymerase chain reaction, which is a technique used to identify or test HIV virus in 8 suspected patient or used for the treatment of cancer. So polymerase chain reaction means even before the microorganism just starts its symptoms on our body, by taking a small, if we are being suspected, we can multiply the concentration, the very small concentration of the microbe, and identify whether that particular microbe is present in our body or not. Then they are used for genetically modified crops for agriculture. What are agriculture? Agricultural modified crops, that is, you will be familiar with the term BT cotton, right? BT cotton, golden rice, all these are biotechnological methods of developing. Okay, golden rice. And golden rice is important. Why? Because it is vitamin A enriched rice. Okay, vitamin A enriched rice, which can cause alleviations in the diseases of the vitamin A, such as night blindness, then color blindness, etc. Then processed food biotechnology is important. Bioremediation, waste treatment, energy production, etc. Okay, you can find biotechnological as the application in all these fields. Clear? Next one. Let me check how many are there. Yes. Okay, let's continue. So these are the different applications of biotechnology. That is, biotechnology is used in medicine. 1 minute. Biotechnology is used in medicine. Biotechnology is used in diagnosis. Biotechnology is used in vaccine, in waste disposal, in agriculture, and in the case of processed food. Is it clear? So medicines, diagnosis, vaccine, processed food, agriculture, and waste disposal, you can find biotechnology. Clear? So let's continue. So in this chapter, we'll be discussing about what agriculture. Okay. So I hope the slide is visible. Okay. So we are going to discuss about the major research areas of biotechnology. Okay, the three main critical research areas of biotechnology include providing the best catalyst in the form of GMOs. In the form of GMOs. What is GMOs? GMOs means genetically modified organisms. Okay, genetically modified organisms. Okay, in the form of improved organism, usually a microbe or a pure enzyme. Then second one is creating the optimal condition through genetic engineering for that particular catalyst to act. So the catalyst may be the genetically modified organisms. So you have to provide a suitable conditions through engineering for a catalyst to act. Clear? The last one is a downstream processing, which I have already taken or discussed with you in the last class of biotechnology, which was being applauded. Okay, so in order to purify the proteins or organic compounds, downstream processing takes place only under aerobic condition. Okay, clear? Very important. So sometimes you may ask a question, but it's not an important area of this chapter since we have much other important areas. Okay, so biotechnology in what? Biotechnology, 1 second. Yes, biotechnological applications in agriculture. Okay, so food production can be increased by agrochemical based agriculture, organic agriculture, and genetically engineered crop based agriculture. So crop based agriculture, organic agriculture, and agrochemical based agriculture you have. Clear? So all these are different areas of how food production can be increased. So here we have another method that is known as the green revolution. So green revolution helps in increasing the food production triple time. You have what? Helped in increasing the food production thrice. And the father of green revolution in India is M.S. Swaminathan. M.S. Swaminathan. M.S. Swaminathan. Clear, everyone? Very important. General information, okay. GI, general information. Okay, so these are the research area, not the research areas. How we can increase the food production by using agrochemical industries, based agriculture, organic agriculture, and crop-based agriculture. Clear? So let's continue with the next one. Green revolution, very important, 100% direct MCQ for NEET. Okay, so I'll be just starting a new series since for my NEET batches. That will be the MCQs. I'll be discussing all the MCQs of the previous year, which will be useful for both NEET 2020 batch or the future batches also. From all the questions, AIMS, AIPMT questions, or state-wise questions, or whatever, NEET questions, whatever it will be, I'll be discussing a few questions as per the time limit allotted. Okay, so let's back to our class. Green revolution. Okay, I have already discussed the green revolution, which helps in tripling the quantity of food. Okay, so production of food many-fold. It was actually 3. Triple the product, triple the food quality by using better management practices and use of agrochemicals and fertilizers. So by using these fertilizers, pesticides and agrochemicals, the food production was not possible. Okay. So to overcome this, we use genetically modified crop or GM, genetically modified plants. So plants, bacteria, fungi and animals whose gene have been altered by manipulation is called GMOs or genetically modified organisms. Is it clear? Understood everyone? So the plants or animals or whatever, whichever organism may it be, so whose genes have been altered by manipulation, you will be calling it to be as GMOs or genetically modified organisms. Clear? Next slide. So there are many applications of GM plants or genetically modified plants. What are they? So first one is crops are made tolerant to abiotic stresses. The very first point, they have been made tolerant to abiotic stresses. So this is how the DNA sequence, okay, this is how the DNA sequence are being inserted. After taking the DNA, but it will be inserted not like this, just pressing and inserting. No, okay. It won't be inserted like that. It will be just made of solutions like this. Okay, solutions like this. And it will be inserted into certain fruits. Okay, fruits. In order to make it ripen, for the ripening of fruit. You may misunderstood it with what? You may misunderstood it with the pesticide. Similar as such, the DNA is also being inserted exactly like this. So, GM plants are tolerant to abiotic stresses. What are the abiotic stresses? Which are the main abiotic stresses against which the crops are tolerant? Yes, correct. That is called then drought, then salinity or salt, then the temperature, heat. Clear everyone? Yes. Then reduce reliance on chemical pesticides. Okay, they themselves, the genetically modified organism themselves have the ability to resist. For example, BT cotton. I'll show the images, so I'm not writing now. Not to make this beautiful images slide. Very dirty, right? So I'm not going to just write everything. So for example, the BT cotton, then the tobacco, disease resistance in tobacco, root, etc. are examples of this reduced reliance on chemical pesticides. Then reduced post-harvest losses. Okay, reduced post-harvest losses. Then they have the increased efficiency of mineral usage by the plants, thereby increasing the fertility of the soil, thereby increasing the fertility of the soil. Then vitamin A enriched rice, that is, you will be calling it as to be what? The golden rice. Okay, the golden rice. They increases the nutritional quality. Okay, so next slide. Is it clear everyone? If you have any doubt, please do it in the comment section. So I'll take a separate class for that and we'll upload that. Okay. The very first topic that you can expect nearly guaranteed MCQs for your NEET. Either for NEET 2021 or 2022, you will expect, we will get a question from this particular session. Pest resistant plants. Clear? You will get a question, sure. So, pest resistant plants reduces the amount of pesticides. They have reduced reliance on the pesticides. Bt toxin, okay, Bt toxin is produced by a bacteria known as Bacillus thuringiensis, Bacillus thuringiensis, okay, produces, which is a bacterium. It produces a specific toxin, a toxin that is Bacillus thuringiensis, that is Bt toxin is obtained from that particular bacteria, and this particular bacteria produces a protein known as cry protein. Cry protein or crystal protein. You will be calling it as to be what? Crystal protein. Okay. So these BT toxin have been, okay, this BT toxin, okay, so this BT toxin gene have been cloned, okay, this BT toxin gene has been cloned from the bacteria, okay, has been cloned from a bacteria and has been expressed in plants, okay, this cloned gene has been expressed in some plant. I'll just provide you the mechanism very diagrammatically, which helps you to understand the concept very clearly, okay. I'll just make, okay, to provide what? Resistance to insect. To provide resistance to insect. Clear? To provide resistance to insect and without the need of insecticide. That is, they actually created what? A biopesticide. Okay? They created what? A biopesticide. So the examples include, what are the regular examples? The examples include BT cotton, BT corn, BT rice, BT tomato, BT potato, BT soybean, etc. Okay, BT corn, BT corn, BT cotton, BT brinjal, BT brinjal. Also, you can write, okay, brinjal, BT brinjal, and BT tomato. Clear? All are being provided in diagrams. Clear? Not diagrams, in pictures. So let's move with the next one. So BT cotton. Okay, BT cotton. And no one has asked me what is this BT, right? Is it, children? So what is this BT? BT stands for Bacillus thuringiensis. Okay, the bacterium Bacillus thuringiensis that produces a crystal protein or cry protein, and that is the BT. Clear? The gene of the bacterium from which the particular toxin has been what? Encoded. That is that. Okay, so this BT plants have produced certain protein that will kill insects like lepidopterans, such as lepidopterans, then coleopterans and dipterans. Okay, will kill certain insects such as the lepidopterans, dipterans and coleopterans. Okay, so what does this lepidopterans include? The lepidopterans include which all things? The lepidopterans are armyworm, then bollworm or the budworm, tobacco budworm are examples of what? Tobacco budworm are examples of what? Lepidopterans. And what is coleopterans? Coleopterans includes a beetle. Okay, armyworm, budworm, that is coleopterans. Okay, coleopterans. Not the butterfly, it's actually some kind. I have just shown you the image. Okay, comes under the category of lepidopterans. Then coleopterans is the beetle just below, right? Isn't it? Coleopterans include the category of beetles that attack the plants. And dipterans includes the flies and the mosquitoes. Includes the flies and the mosquitoes. Is it clear? Look at this beautiful images of the vast variety of organisms that is available to us in our environment, isn't it? So the next one, let's move in detail about the BT cotton. So Bacillus thuringiensis produces a crystal, okay, crystal protein that contains an insecticidal toxic protein. So this protein, sorry, okay, this protein is present in bacterium. So this particular very important 100% MCQs, or I'm guaranteeing you, guaranteed MCQ, my signature. Guaranteed MCQ may expect this particular question for your board exam as well as for your, mainly for your NEET, you will definitely get a question. Okay, this particular statement you mark it and study because it is a statement from which the questions are being asked. So the toxic protein present in bacterium as inactive protoxin. But as soon as the insect ingests the inactive protein, due to the alkaline pH of the gut, it gets converted into an active toxin. It binds to the surface of the mid-epithelial cells and create pores that causes a cell lysing and cell swelling and eventually causes the death of the insect. This one paragraph, this is actually a very single statement. Look, I have separated it only with a comma. Okay, it's a very, very important statement. Direct from your NCRT itself, you may expect a question. Very, very important. You may expect. Okay, this particular thing is very important. Very, very important. And again, triple marking it with a highlighter. Very important. Okay. So I'll just explain you with the help of a diagram, and this is a diagram. Okay. Yes. So this is that leaf of the BT cotton. Okay. This is the leaf of the BT cotton. You suppose leaf of BT cotton. Clear? And the larvae or the caterpillar ingests the BT spores and the cry protein. So this is a cry protein. This is the cry protein. And this is the BT spores. So this both cry protein and the BT spore are ingested by this particular larvae or the caterpillar. Clear? Is it clear? Then in the larval midgut, okay, in the larval midgut, the proteolytic digestion of protein releases a crytoxin which bind to the epithelial receptors. Okay, look, this is a cry protein. This particular yellow colored triangle, I'm marking, highlighting it with a red. Okay, so this particular protein is the cry protein or the crytoxin binded with the cry protein with this red outline. Clear? Okay, so this particular thing after reaching the gut, okay, reaching the gut, what binds with the epithelial receptors, and this is due to the alkaline pH of the gut. Clear? So this particular thing, this binds with the receptor binding site, epithelial receptor binding sites, and what happens, this toxin binding causes a cell lysis due to osmotic cell lysis. This you will be calling it as the osmotic cell lysis, destroying the barrier to the body cavity. So this is the gut. Okay, these are the gut of the human, the body, gut of the body cavity, and this causes the entry of all other microbes, So these are the microbes. So what happens? This particular attachment of the crytoxin to this particular what stiff cells or stiff cell causes a lysis or what swelling of the cells and allows all the microbes to enter inside the body. So this is the decoding process. Okay, clear? Is it very clear? If you want, please take a screenshot of this particular slide, because diagrammatic representation will definitely be a good idea in order to make you understand the topic. Clear? So I have some other diagrams also, I think. Yes, have some other diagrams. Yes, this is how you know, look. So this is a BT toxin and these are present in the leaf. Okay, they are present in the leaf. So when this caterpillar or the larvae just feeds, this cry protein will enter into their body. Okay, they will definitely enter into their body, and they will reach the epithelial midgut. And what happens is that they will bind with the epithelial lining cells. They will bind with the epithelial binding cells. Okay, and causes a what that stiff cells become weak like this, and ultimately entry of all the microorganisms into their body. The receptor cells will be absent. So finally it causes a death of the particular insect or whatever it is to be, it causes a death. Clear? So I think one more. Yes. This is how this procedure very clear. So this is the bacteria Bacillus thuringiensis. Okay. Okay, sorry. Okay, Bacillus thuringiensis, a gene will be inserted. So this is a particular gene present in this particular bacteria. So that gene is taken out. And this BT gene is inserted into the crop plant. So this is a crop plant. Inserted into the crop plant. The crop plant is affected by the cotton borera. The crop plant is affected by, look how beautifully the diagram is being represented. The bacteria, not the bacteria, the organism is just coming with a knife and a fork to just eat the plants. It's just shown for you to understand. But this plant is strongly resistant, right? So this plant is resistant to all the attack. Clear, this plant is resistant to all the attack. It's very strong plant. So as soon as it eats the leaves of the plant, it will finally result in the death of the pest. Okay, it's pest. Clear? Next one. So this is a procedure. Since this topic is very important, I'm taking again and again the classes from this particular thing itself. Okay, children. So let's continue. Only a small portion is being left out. So the BT in inactive form, the Bacillus thuringiensis toxin is sprayed into the crops in the inactive form. Then they are eaten by the insect. And this toxin, as soon as it reaches the gut of the insect, I have shown you, right? So as soon as it reaches the gut, oh my God. As soon as it reaches the gut, it causes a lysis of the cell, right? It causes the lysis of the cell. Okay, back. So please take a snapshot of this particular slide. This will be very, very useful for you guys because they will be giving these things in a mixed sequence and be asked to arrange you. Okay, very important one. Direct questions you can expect. Seriously, direct questions. Okay, so the BT gene is being transferred into this plant, and on the pest eating the leaf, European corn borer, study this term, European corn borer, BT cotton, and finally causes the death or the collapse or lysis of the cell. The term is lysis. Okay, finally causes the death of the insect or the swelling of the gut of the insect. So we have come to the end of this topic. Next one. So this is also very important. The gene from Bacillus thuringiensis. Okay. Gene from Bacillus thuringiensis are incorporated into several plants. Okay, for what? Like cotton. Sorry. Okay. Like cotton, then maize, rice, etc. So this toxin is coded. This toxin, so very important, okay? So this toxin is coded by a gene known as CRY. The protein coded by the gene CRY1AC, and actually it is a mistake, okay? Actually this is a mistake, I'll just correct it for you, okay? That's a slight mistake. It's not CRY1AB, it's CRY1AC. Cry1AC and Cry2AB. Cry1AC and Cry2AB control the cotton bollworm. Okay, control the cotton bollworm, whereas Cry1AB control the cotton borer. Look, this is a cotton corn borer. Okay, not cotton borer, it is corn borer, and this is the cotton bollworm. Okay, how beautiful the images are. Please look. Hope you all will enjoy the session very well. Okay, so in spite of this enjoying the pictures on the slide, please do share it with your friends. Don't forget to like, share, and subscribe to our channel. Okay, very important session. Okay, next topic, that is a pest resistant plants. Certain nematodes. Okay, certain nematodes like Meliodigine incognitia. Meliodigine incognitia infects the root of the tobacco plant and causes reduction in yield. Clear? So this infestation, the infestation of these nematodes are prevented by a process known as RNAi or RNA interference, that is the silencing of the mRNA. So RNAi is present in all eukaryotic organisms. RNAi is present in all eukaryotic organisms. By silencing a specific mRNA, how? By producing an mRNA, we will be producing a complementary dsRNA. And thereby they will complementary RNA and what? They will just bind together to form a double-stranded complementary RNA, dsRNA. So, due to a complementary dsRNA molecule that binds to it and prevents the translation of mRNA. Is it clear? Is it important? Yes, it's very important. Clear? So, the source of the complementary dsRNA from the viruses having RNA genome through RNA intermediates. Okay. Then the nematode specific genes were introduced into the host plant using agrobacterium vectors. Is it clear? Agrobacterium, very awkward to see, right? So this is the thing which you have also seen, which may happen in your plants. Okay. Mainly happens in the roots of tobacco. So this parasite could not survive in the transgenic host expressing the interfering RNA. Is it clear? Not so important. Not so important also, I cannot say it's important, but no more concept, just like how the process takes place. As just like what happens in case of the last one, right? Bacillus thuringiensis. We don't have a detailed explanation for this particular nematode resistant plants. Clear? So this is a diagram. Okay, this diagram is given in your NCERT also. If you look, this is a host plant generated dsRNA triggers the protection against the nematode infection. For example, you can see the B part shown there is a transgenic plant root 5 days after deliberate infection of the nematode after protection through novel mechanism. Clear? And this is the infected one. Okay, roots of a typical control plants. Okay, not infected, roots of a typical control plants. So next one. So we have come to the end of this wonderful, super amazing session. So once again, thanking you all for your patient listening, and please don't forget to like, share, and subscribe our channel with your friends. Because, okay, please don't forget to like, share, and subscribe our channel. So once again, thanking you all, and it's bye from your favorite Hearts exclusive channel till we meet with the next lecture of this particular chapter. Okay, so please don't forget to click the bell icon so that you will get the latest notifications about the upcoming series. Okay, so thank you all.